All right, so we're ready to continue our discussion of membrane proteins. Remember, there are two different kinds, right? You can have your integral proteins, those that are actually embedded in the phospholipid bilayer of the membrane, and then you also have the peripheral proteins, which are just attached to the side of the membrane. Now, irregardless of what kind of proteins we're looking at, either integral or peripheral, the membrane-associated proteins can have a variety of different functions. And so over the next two slides, we're going to go through 6 of the different functions that membrane-associated proteins can have. So in this slide, we have our first 3. So the first example, a lot of membrane proteins can actually be transport proteins. And these guys are going to be the ones that help the cell to get materials across that hydrophobic core of the plasma membrane itself. We're going to spend some time talking about different examples. Some of them are carrier proteins. Some of them are going to be channel proteins. Irregardless, again, they shuttle material across the membrane. Our second function is enzymatic activity. So let's say a cell needs a particular chemical reaction to take place near the plasma membrane. What better way to localize that particular chemical reaction at the membrane than to take that enzyme that catalyzes that reaction and essentially just stick it into the membrane so it can't travel any other place within the cell. Number 3, the third function of the membrane-associated proteins is going to be signal transduction. And this is something, again, that we've talked about previously when we were going through just protein functions in general, right? So a lot of the proteins embedded in the membrane can function as receptors or these mailboxes, for example, for signaling molecules like hormones. The signaling molecule will bind to the receptor molecule here on the outside of the cell, and then the receptor generally is going to change shape, and it'll interact with other molecules on the inside of the cell to get that signal or that message into the inside. And so the cell can then respond to that signal as it goes through a variety of other processes. All right, our last 3 functions of membrane proteins. Number 4, cell-to-cell recognition. This is particularly important, and we're going to come back to this idea actually on the next slide to sort of reinforce it. But oftentimes cells need to be able to communicate with each other. This is especially true for cells of the immune system. So certain cells travel through your body and sort of act like patrols and make sure that there are no invading particles that could potentially cause infection. And so these immune system cells have to have the ability to differentiate between what is your cell and should be left alone versus what is an invading cell which should be degraded. One of the ways that cells can express themselves or say, hey, I'm a skin cell or I'm a muscle cell is by actually expressing certain carbohydrates on the surface of the membrane. And so in this particular image here, you see here's the membrane of one cell. It has a protein embedded in it, and that protein has a carbohydrate attachment added to it. This is an example of a glycoprotein, a protein with carbohydrates on the surface. And on the opposite side, here's where you're going to have that cell that is essentially trying to recognize that other cell with a glycoprotein, right? So this particular image is showing you that cell-to-cell recognition. One cell is using its membrane-bound protein to interact with the glycoprotein that's embedded in a neighboring cell membrane. All right, in the middle here we have intracellular joining. So proteins are oftentimes used to attach 2 cells together. This is something that we saw, for example, when we were talking about animal cell junctions, the desmosomes. That's a really good example where you have many proteins involved in the creation of the desmosome, and those proteins interlock between the 2 cells and help to keep those cells tightly attached to each other. And then the 6th function, again, something we took a look at the very end of chapter 6, and this is the use of those membrane proteins to sort of attach the extracellular matrix to the cell, and then also act as a place of attachment for those cytoskeletal fibers on the inside of the cell. So it's almost like you have a nice little continuation here of fibers from the outside of the cell through these membrane proteins to the inside where they attach here to the actin microfilaments on the inside. So clearly, there are a lot of different functions, even for those specific membrane-associated proteins. All right, to go back to our fourth function that we just talked about, right, that was cell-to-cell recognition. This slide will reinforce the importance of these carbohydrates on the surface of the cell. On the previous example, if I just go back for a second, right, previous example, this was a protein that was embedded in the membrane that had the carbohydrate attachment added to it. Proteins aren't the only things that can have those carbohydrates added to the cell surface. If you take a look at the little figure over here on the right, you'll notice that yes, we have purple proteins here with these large carbohydrate attachments. There's another one back here, the carbohydrate. Now up in front though, we have another little green carbohydrate here, and this guy is actually attached to a phospholipid instead. So not only can you have proteins with these carbohydrates added to the surface, this forms glycoproteins, but you can also have lipids that have those carbohydrates added to the surface, and those are going to be called your glycolipids. Now, glycoproteins are much more common in a cell, but glycolipids do also exist, and both types of macromolecules with these carbohydrate attachments function in the cell-to-cell recognition, right? Identifying what kind of cell that particular carbohydrate is attached to. My usual analogy is, you're walking down the street and you see somebody in scrubs, or you see somebody in a police uniform. You kind of have a pretty good idea of what that person does, as their day job, right? So it's kind of the same thing for the cells. The cells put on a uniform, right? These carbohydrates on the cell surface, and that allows other cells to identify what kind of cell that is. All right, so we've gone through structure of the membrane. We've talked about fluidity and the lipids. We've talked about different kinds of proteins and their sort of functions and the way that they can be associated with the protein. Now it's time to take the membrane as a whole, right, and talk about its function. How do all those components come together and allow this boundary of a cell to be a selectively permeable barrier? So we know that a cell has to exchange material with its surroundings, right? Nutrients have to come into the cell, oxygen has to come into the cell, and then the cell uses that material, and as a result of the chemical reactions that take place inside, it produces waste. So it has to get rid of that waste, it has to get rid of carbon dioxide, for example. So many materials need to be passed across that plasma membrane, and yet the plasma membranes are selective, so not everything is allowed to pass across easily. So let's take a look at this particular animation, sort of introducing selective permeability of membranes.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
All right, so that video was a good little introduction identifying that, okay, there are certain substances that easily pass across the membrane on their own and others can't. And this is really all a result of whether or not that substance is hydrophilic or hydrophobic. So let's take a look at these two different kinds of substances and how easily they might be able to pass across that membrane. So things that are hydrophobic don't like to be with water. Or molecules that are non-polar, things like hydrocarbons, for example. They can easily dissolve through that lipid bilayer. They're not afraid of those hydrophobic fatty acid tails of the phospholipids, so they'll pass through really easily. So things like lipids will get across pretty easily. Or really small non-polar molecules like oxygen or carbon dioxide. They have a very easy time getting through that phospholipid bilayer. On the other hand, any molecule that is hydrophilic, likes to be with water, those types of molecules are going to have a very difficult time crossing that hydrophobic core of that phospholipid bilayer. So things like proteins or water itself sometimes has difficulty getting across those fatty acid tails. All right, so anytime actually that we're discussing the movement of molecules across a membrane, there has to be some sort of push, right? There's some sort of desire for those molecules to get to the other side. So this takes us to a discussion of the process of diffusion and the process of osmosis as well. So let's start out with diffusion. Now diffusion in general, not even in the context of a membrane, is defined as the tendency for molecules to spread out evenly into the available space. Right, so in the animation at the bottom there, I'll play it in a second, but you can see that those green spheres are sort of located on one side of the blue box, right? So there's an uneven distribution. If we let those molecules sort of hang out and sit just by random movement, again, because they have that thermal energy, that kinetic energy, and they're bouncing around, right, they're going to end up spreading themselves out within that blue box until their concentration is essentially even throughout that blue box. All right, so let's take a look at the animation.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Okay, so again, you saw how those green spheres then ended up diffusing through the blue box and spreading out to reach that sort of equal distribution throughout the given area. And this can happen with any molecules, right? So for example, you spray perfume, right, in a small room. Eventually, those perfume molecules are going to diffuse through the air and they'll make their way from your bathroom to your bedroom, eventually maybe even to your kitchen, right? And so that's, again, just a great example of this process of diffusion. Those perfume molecules want to spread out into whatever space is available to them. And this process of diffusion also, as the animation showed you, happens across a plasma membrane. Right? So the molecules are going to want to move from an area of high concentration to an area of low concentration. So if the concentration of molecules is different on two sides of a membrane, that'll be a really good push for molecules to want to get it to the other side. They want to go from where they're packed in really tight to where they have more space to spread out. So let's take a look at an example. This example is using essentially, let's say, a beaker. Right? So I have a beaker of water here, and I've separated the two halves of a beaker with a membrane. And let's say I add a dye to the left-hand side of the beaker. It's an orange dye. You can see all the orange dye molecules there. Now, the membrane that I use to separate the two halves of the beaker is permeable to the orange dye. So as the orange dye molecules are bouncing around in here, some of them will pass across that membrane and they'll be bouncing around here on the right-hand side. Right, so you can see now that as we let this sit longer and longer, eventually enough of those orange dye molecules will move to the other side and they will all be spread out until the system essentially reaches what is called equilibrium. They're at equilibrium, there are going to be equal concentrations of the dye molecules on either side of the membrane. Even though, if you think about it, the dye molecules are still moving at equilibrium. They're still bouncing around. But for every one dye molecule that moves to the right-hand side, another one is going to cross to the left, maintaining that equilibrium or that equal balance and concentration of the dye molecules on both sides of the membrane.